So today's talk is about light scattering techniques. There's quite a bit to go through, and I apologize if I don't keep within the hour, but I will do my very best to do that. And the talk's divided into these sections here. I'm going to cover the basic theory of light scattering. I'm going to talk predominantly about static light scattering and its implementation in a technique called SEC moles. And then at the end, I'll talk about dynamic light scattering and then very briefly mention some other uses of scattering that might be useful in your work. So light scattering has quite a strong connection with Cambridge and also with the LMB, in that the LMB was born, if you like, at the Cavendish labs down in town, where Max Perutz and John Kendrew started work on crystallography in what was essentially a shed behind this, the main Cavendish labs. And the Cavendish labs were built in 1874, and the first Cavendish professor of experimental physics was James Clerk Maxwell, and he is very famous for his series of equations that describe electromagnetic radiation. And within those, you find that light is composed of oscillating electrical and orthogonal magnetic vectors, or that's the description. And when you consider polarized light, this polarized light has a single electrical and a single magnetic field. And the intensity of light, which we're often interested in, is proportional to the square of the amplitude of this electric vector. And it's the interaction of this electric vector with matter, which is the major contributor to light scattering. After James Clerk Maxwell, the second Cavendish professor of experimental physics was this person called John William Strutt, who was also a lord, and known Lord Rayleigh was who he was, and Rayleigh formulated or investigated and proposed how it was that electromagnetic radiation generated scattered light, or further electromagnetic radiation scattered from particles. And what he proposed was that the electric vector in electromagnetic radiation induces oscillating dipoles in any matter where there are charges that can be separated, and this is known as polarizable components. And these oscillating dipoles, which are induced by the electromagnetic radiation, re-emit light at the same frequency, and so this is a form of elastic scattering. The wavelength of light is the same that's emitted, and this is so-called Rayleigh scattering. And if we induce scattering with polarized light, so there's only one electric vector, the scattering is predominantly in a plane which is perpendicular to that of the incident light. So here you can see the electric vector of the polarized light, incoming light, and this is the emission of the scattered light. And the intensity of scattering reflects then the polarizability of your substance. And polarizability is also reflected in the refractive index increment of a substance. This being how much the substance changes refractive index per change in concentration. So the more a substance affects refractive index, the more polarizable it is. So how is this useful to us? Well, one of the major uses of light scattering, certainly here at the LMB, is that if we consider a large particle, it will have many, many dipoles in it. And if these are located within a single particle, they will emit light coherently, whereas the dipoles in particles which are independent will emit light which is out of phase. So when you then think about the intensity of the coherent light coming from the individual particle, we have to sum up the electric vectors and then square them to get the intensity of the light, so-called square of the sum. But the intensity of incoherent light from these separated particles, which are out of phase, is just their intensity summed, so the sum of the squares. So if you have an identical level of polarizability in these two sets of particles, then the intensity of scattered light is going to scale very strongly and non-linearly with particle mass, as shown here. And when we think about static light scattering and also dynamic light scattering instrumentation, what we do is use plain polarized laser light sources, and we measure the intensity or the fluctuations of this scattered light in a perpendicular plane to the polarized light that we're inducing the scattering with. So usually for biological work, most instrumentation has longer wavelengths so that it avoids any potential absorption from biological material. And so the LMB instrumentation we have, which is from a company called Wyatt, in terms of their scattering, they use a 658 nanometer laser. And particles that are much smaller relative to this wavelength tend to scatter light equally in all directions. When we say equally, they actually don't scatter light equally in all directions, but we're unable to really measure the variation as a function of scattering angle. So these smaller particles appear as isotropic, so-called isotropic point scatterers. As the particle size increases, and in the case of a laser, which is around 650 nanometers, this corresponds to about 10 nanometers in size, you get a loss of coherence from individual particles, and this tends to produce measurable attenuation of the scattered intensity at any angle above 0 degrees. And this is due to intramolecular phase shifts of the scattered light. And by measuring and fitting the attenuation that we see, we can also extract a parameter, which is the root mean square radius of a particle. This is also commonly known as the radius of gyration. If you look here for a laser of our wavelength, a particle which has a root mean square radius of 20 nanometers produces about 5% attenuation of intensity over 180 degree angle. So this is reflecting the fact that actually particles which are smaller than 10 nanometers, they will attenuate the scatter, but to a level that we're really unable to quantify. So what is the root mean square radius? Well, the term radius of gyration gives some sort of implication that there's a dynamic component in this measurement. But in fact, what the root mean square radius is just a measured radius which reflects the mass distribution within a particle around the center of mass. And this mass distribution is weighted by the square of the distance from the center of mass. So this is the formula you would use to calculate it. And the easiest way of just visualizing that is if you have a hollow sphere like a ping pong ball, then the radius of gyration will correspond to the physical radius of this particle, because all of the mass is equally distributed from the center of mass. When we have a solid sphere like a billiard ball of uniform density, then the physical radius, the diameter of the radius of this ball will be larger than the radius of gyration, because some of the mass is nearer to the center of mass. And you can develop other relations for random coil or rigid rods in terms of the relation to the physical size and the radius of gyration. So then after Rayleigh described the phenomenon of scattering and how this is occurring, then Gustav Mie produced the solution to Maxwell's equations, which models the attenuation in the scattering phase function and includes information about the particle radius, the wavelength of light, and the scattering angle. So particles which are much smaller than the wavelength appear to be isotropic point scatterers, and they scatter light uniformly in all directions. But particles that are closer to the wavelength of scatter will tend to scatter predominantly in the forward direction. And if you graph out the intensity of scattering as a function of angle, as this so-called size parameter, which is 2 pi r divided by the wavelength, you see here when the size parameter is around 10, the radius is approximately equal to the wavelength of light you're interrogating it with, and you'll see that the intensity of light is universally in the forward scattered direction. So typically we are measuring with particles that are much smaller than the wavelength, isotropic particles, and then we get a uniform or a quasi or essentially constant level of scattering as a function of angle. So this brings forward a few interesting observations. So the Me theory will describe all scattering, but unfortunately the mathematics of this is extremely complex and horribly messy, and this particularly applies in this regime when we think about this size parameter, where the radius of the particle is similar to the wavelength, so-called Me regime. At extremes of small or large particle size, the whole analytical description of scattering becomes a lot simpler. And this is due to either the dominance of the radius or the wavelength in this relationship here. So most biological work that we're doing is conducted using near infrared lasers, and therefore for particles that we're interested in, which might be between 1 nanometer and say 50 nanometers, then the scattering is predominantly in the Rayleigh regime. And analytically, this is a lot more simple. You can also see here that if you wanted to look at, for example, much larger particles like raindrops, you would need to use a much, much longer wavelength if you wanted to be in the Rayleigh regime. And when you see these maps of rain on the weather forecast, they're coming from a radar system, which is a pulse followed by an echo back from the scattering object. And in that case, if you had forward scatter, you would obviously never get your signal back. So to work or to be able to collect data back from the direction that you interrogated the particles with, you need to be in the Rayleigh regime so that scattering is in over all angles. And therefore for rain radar, you use a much longer wavelength here in the centimeter regime. So we can come up then when we're working in the Rayleigh regime with a more simple and analytical description of Rayleigh scattering. And this is related to this size parameter between 0.2 and around about 0.002, which corresponds to radius of gyration between 25 and 0.2 nanometers for the laser wavelength that we have. And within this relationship, we have a produced level of scattering above that of the solvent at any particular angle. We have here a bunch of optical constants, which include the refractive index of the material we're working in, Avogadro's number, the wavelength. Note that this reciprocal wavelength means that there's a strong wavelength dependence of the intensity of scattered light. We have here this refractive index increment of the particle we're interrogating. And then within the equation, we have the molar mass of the particle. And this is what we're interested in measuring predominantly when we do light scattering. Here we have the concentration of the material we're looking at. Then we have this angular term, which is reflecting the attenuation of scattering as a function of angle. And then here in this final term, which includes mass and concentration and the attenuation factor, we have this other parameter called the second virial coefficient. I'll describe that later on, but normally we can generally ignore that, or certainly ignore the contribution of this term, as it tends to be very small, and then therefore this term in the square bracket is equal to 1. So light scattering in practice then, how do we actually use it and measure it and analyze the data we get? So within the LMB, I've mentioned already, we're using this wire instrumentation to measure light scattering. We have a multi-angle light scattering detector, which has 18 angles in it, and these are arrayed between 22 and 150 or so degrees of angle, and the wavelength is 658. Using this detector, we can measure the intensity of scattered light. We also use a differential refractometer, and this allows us to measure the dN by dC. So we need that parameter, the polarizability of our particle within the equation. If we already know the dN dC, and for proteins, you'll see it's very well established and known, then actually we can use the refractive index change from our substance to determine the concentration, because here we have dN by dC is a change in refractive index per change in concentration. So refractive index can be used both to determine the polarizability, but also as a measure of concentration. We can also use other types of concentration detector, classically UV absorbance, say at 280 nanometers for proteins. And when you combine these two measures of concentration using dual concentration methods, it can act both as a cross-check to confirm that you're getting the same molar mass by either concentration source, or also in more sophisticated types of analysis known as conjugate methods. And I'll describe those briefly in a moment. So differential refractive index is measured in this differential refractometer, and has been well established for many years, that the refractive index increment, the refractive index increment for proteins has a pretty well universal value of about 0.19 for a 1 gram per mil solution. And so we can quantify the concentration of proteins from differential refractive index measurements. This is particularly useful if we want to study particles that don't have any UV absorbance. So that might be proteins without any aromatic residues, or if we want to look at other biological materials like carbohydrates or or detergents or whatever. So if there's no UV absorbance, we can still get the concentration using the refractive index. Proteins are pretty highly polarizable, and therefore they scatter pretty well. This is a fairly high number for a refractive index increment. When you think about the structure of proteins, of course, yes, they're full of polarizable groups within them, in the backbone and in the side chains. Just as an instrumental point, if we're going to be measuring proteins or samples around about mgs per mil, then the change in refractive index for 1 mg per mil will be 0.19 times 10 to the minus 3 of a refractive index unit. And therefore, when we're measuring differential refractive index, we're trying to work at pretty low levels of refractive index change. And this is quite technically challenging in that the background solvent, or our buffer refractive index, is going to have an RI around 1.3, since it's predominantly water. And therefore, the instrument uses a split cell to offset the background refractive index of the solvent. So if we were then going to measure the intensity of scattered light, we could consider doing this as a so-called batch measurement. When I say batch measurement, this just means put the sample in some sort of measuring holder like a cuvette and put it in the machine, measure the intensity. But we've seen already that scattered intensity is proportional to mass and in this non-linear manner. So low concentrations of large particles will significantly bias the measurement, and therefore your apparent mass will appear higher than it really is because of the large intensity contribution from even low levels of small particles. So if you want to do, I'm sorry about this, it's the slide changing. So if you want to do batch measurement, then the material you're using must be highly monodisperse and stable and not prone to aggregation. If it isn't, you can filter the material with very fine filtering devices with very small apertures. This may well help, but in a way, this is kind of hiding the polydispersity that you might be interested in when you're making your measurement. So in fact, if you could fractionate material prior to measuring this, so rather than doing a batch measurement, you worked with fractionated material, this would be somewhat more optimal. And this is exactly what we do with the implementation in so-called SEC moles. So this is just size exclusion chromatography with multi-angle light scattering. And the way the system is configured is we have some sort of auto sampler or way of injecting the sample. We have a standard size exclusion column on the system, normally a GE column, and then we fractionate particles according to their hydrodynamic radius. And these particles then pass through a series of detectors, a UV detector, which is normally part of the chromatography system, then the light scattering detector, and then the refractive index detector. What we end up with then is a chromatogram, and one of the signals is the UV, another signal, all these lines down here are the light scattering, and then this blue one is the refractive index. And every single point in our SEC moles chromatogram can be used to analyze to give us a mass of a particle and the radius of gyration, providing that radius is a bit above 10 nanometers. So this is typically what the data look like, and there are several things that you can see. This measurement was done with 100 microliters of 1 mg per mil BSA, so it's using 100 micrograms, and it just used a standard analytical SuperDex 200 column. I have to say that if you use the SuperDex 200 increase columns, which are now more commonly available and pretty well universally used, then the resolution of these species, this is a monomer of BSA and this is a dimer, the resolution is hugely improved, and so you get even better separation of the monomer and the dimer. But in terms of more general features, we can see when we look at the light scattering signals here, which are all these lines, there are potentially 18 angles overlaid here. We can see at the column void, we can see significant signal. And this, when we then look at the concentration signals like the UV or the refractive index, we can't actually see really any significantly measured concentration. This is the exact problem that I pointed out with a batch measurement, that despite the fact you have extremely low levels of these larger particles, the intensity of the scattered light is almost as great here as the intensity that you're seeing from the monomeric species. Here at the end of the separation, refractive index is particularly sensitive to changes in solvent. And obviously, often your sample might be in a slightly different buffer when it's injected onto the column, and different from the running buffer. And so you tend to see this solvent front as a large change in refractive index. In terms of UV and light scattering, there's no signal at all. So this large deflection is due to solvent differences. So how do we analyze this data? Well, just to point out that here, if you look at the UV signal, you'll see that there are a series of points collected, say, every 1 second. And for every single point within our chromatogram, we can generate a so-called Debye plot, which is plotting out the intensity of scattered light as a function of angle. And on the y-axis, we have this K star times concentration divided by the intensity of measured intensity of scattered light. So because this is a reciprocal of intensity, this is high intensity and this is low intensity. So what we do is we use the y-axis intercept of this Debye plot, which is then extrapolated to 0 angle, because under those conditions, then this attenuation of the scattering as a function of angle reduces to 1, since this whole term here becomes 0. So we evaluate the mass by extrapolating the intensity of scattered light to 0 angle, and then inputting that into the Rayleigh type equation along with our measurement of concentration. And a Debye plot can be plotted for each data point within a particular region of the chromatogram. The slope of this Debye plot then is reflecting a change in intensity. This is decreasing intensity as a function of increasing angle. So this is the attenuation of the scattering as a function of angle. And therefore the slope of this plot can be used to determine RG. It should be positive because of this being high intensity and this being low intensity. But of course, it's only really measurable in terms of giving us a number we can trust when the particle is above 10 nanometers. So data can be averaged again, or at least this radius of gyration could be determined at every point in the chromatogram. We can average the data just as we can with the mass within different regions of the chromatogram. And there are other methods apart from this Debye type of analysis, the Berry and random coil formalizations, and these may be more appropriate when you know that your particle is very large or you know that it's non-globular. So as I've said several times, we can determine the mass. In this case, this is the molar mass plotted out at every single point within the chromatogram using this so-called Debye plot. And this will generate the mass at different regions within the chromatogram. And so here we can see the monomer of BSA, and here we can see the dimer of BSA. We can either use the data in this way to show it graphically, and maybe we would put somewhere within the text of a paper or whatever what this level corresponded to. But another way of dealing with it is just to average the mass determinations within that region. So when we've defined some region within our chromatogram, we can then average the individual masses that we've determined. And this can be done normally and in the light scattering field in two ways. You have MN, which is the straightforward number average, or MW, which is a weight average. And you'll see here on the right that if you have a uniform series of particles, so-called monodisperse distribution, it doesn't matter if you use the number average or the weight average to average these data, the eventual answer is the same. And therefore the ratio of these two averages is sometimes quoted as an index of dispersity. If the material is monodisperse, the value should be 1. Here you can see polydispersed material, so there are particles of all different sizes. And when we do the averaging using those two different methods, you can see that we get a different number and the ratio would not be 1. But what is this really reflecting? Well, what it's saying is that when we have monodispersed material, all the masses that we determined and that we're averaging across the peak were roughly the same. So that it doesn't matter how we statistically average them, we get the same number. And this really reflects the fact then that the mass is constant within that region of the chromatogram. So this is what monodispersed material would look like: the monomer, the dimer, and the trimer of BSA, where the masses within regions where we have reasonable signals are constant. And this is a highly polydispersed sample, where the mass varies hugely across the peak, and the ratio of this would not be 1. So you have two alternatives in terms of talking about the mass and the dispersity of your sample. One is to use a graphical method of displaying it, and the other is to calculate the statistical mass moments as described. So what are the applications that we have for light or for static light scattering and this SEC moles implementation within the LMB? Well, one of the simplest applications is just to confirm the mass. So we would claim that the determination of mass is fairly accurate. So we would look for an error within a few percent of expected mass, or if we have run a standard, we would look to obtain a mass that was within a few percent of that known mass. It's a relatively quick technique, so it's just a size exclusion, analytical size exclusion run. It takes about 45 minutes. But the important thing is that we're determining mass in solution, and we have a pretty good dynamic range. We can measure over between very, very small particles, although you need pretty high concentrations, up to really quite large sizes of complex, and hopefully obtain similar levels of accuracy. The other thing to point out is that this is totally independent of the elution time from our chromatography system. So when people do size exclusion chromatography, they're often thinking about what's the elution time. So this x-axis, which can be volume or time or whatever, is not used in any way in the analysis. So it does not matter where the material elutes. The mass has been determined by a completely independent method. So here you can see just some examples of some proteins that we ran, where the known mass of these from sequence, and this is the experimentally determined mass, again using small amounts of material. Here's one that's a slightly smaller protein, here's a larger protein. This application works for all proteins because we have this universal refractive index increment that's used to determine the concentration. And for that reason, we can work with any protein. It doesn't have to have aromatic residues. We don't even need to really know what the sequence of the protein is to determine the mass. And because of the size exclusion step, we can work with slightly less than pure, 100% pure samples, although we don't encourage people to bring very crude samples along. It also works if you have genetic fusions like GFP. In this case, you can see that we're able to determine the mass of this dimer here. Then in a monomeric mutant, it was coupled to GFP, and again, we get an accurate mass. And then here it is forming a dimer with the wild type, again fused to GFP. So this is because the laser we're using to interrogate the material is at a longer wavelength. We can also start to look at interactions between proteins. So for tight binding, this is pretty straightforward. We hope that we can look at the mass of the individual proteins, in this case, a ribonuclease, and then its intracellular inhibitor. known as bar star. And then when you mix them together, we form a complex, and the mass of this complex, if this is a one-to-one interaction, is expected to be around 23 kilodaltons, and that's exactly what we get, measured by refractive index and also determined using the UV extinction coefficient, which we calculate based on this one-to-one complex. So this is a way of cross-checking that the mass you obtain when you use refractive index corresponds to the mass of the proposed complex. So in this case, you could kind of argue, okay, maybe this species here is not a one-to-one complex of bar star and Barnase, but actually some weird dimer of Barnase that you form when you put bar star in, or something like that. Because the mass of the complex is 23, and that's, you know, kind of close to a dimer of this. But obviously a dimer of Barnase would have a completely different UV extinction coefficient from a one-to-one complex between these two proteins. So by doing double concentration determination, it's useful for determining stoichiometry. Here's another example of tight binding, looking at this trimeric hexamer or trimeric protein formed here, and then we were interested in its interaction with a fab fragment. Again, you can see that this is then 3 fabs coordinated onto one of these trimers. Again, the UV determination confirms or helps to cross-check this stoichiometry. If we have weaker binding, then what we might see is that as sample concentration varies during the chromatography, so here obviously we have low concentration, and here, and at the top of the peak, we have our maximal concentration. And in some sort of dynamic equilibrium where we have a binding, but at a somewhat weaker level, then what we might see is as concentration is increased, we form more of a higher order species. In this case, it was known that this was a coiled coil. This is the mass of the dimer. This is the mass of the monomer. And in fact, if you do this at a whole series of different concentrations, you see that both you get information by varying the injected concentration, but also within the peak itself, you know what the concentration is from the refractive index measurement. And therefore, you see this variation in mass occurring because of rapid re-equilibration on the column. So eventually, if you get enough data, you get a pretty good ballpark idea what the KD is for the interaction in this case. So if you go beyond proteins, it's quite easy to study other types of macromolecule, in this case, detergents. And you can see here using the detergent UDM, this is known to form micelles with a mass around about 50 kilodaltons and a hydrodynamic radius of around 3 nanometers or so. And these are both very accurately determined over a wide range of concentrations. So once the detergent goes above the critical micelle concentration, it forms micelles of all type. But the interesting thing just to point out here is that although the mass and the hydrodynamic radius of these samples are all the same because they're micelles, the number or the concentration of micelles passing through the detectors is increasing as we have more and more UDM in the sample. So potentially this gives you a way of quantifying what is the percentage concentration of detergent. And this has proved to be of interest to people who are looking at membrane proteins and trying to crystallize them. And as they concentrate the protein, they're not really sure how the concentration of the excess detergent micelles is varying during the concentration process. So then we can go beyond a simple, straightforward SEC moles measurement where we can use one detector or the other And if we use both detectors, we should, in principle, get the same number. When we think about proteins that are, or particles that are a mixture of different biomolecules, so in this case, a glycosylated protein, because during the measurement, we have two signals, one is RI and one is UV, and both of these will give us concentration. Then when we have a modified system, we can determine the complex, the extinction coefficient of the complex, because it becomes the weighted sum of the components factored by their individual contributions. So if we use this so-called conjugate analysis, the mass of the protein and the modifier, and then the mass of the total complex can all be determined simultaneously. To do this, we would need to know the refractive index increment of the protein, the refractive index increment of the carbohydrate modifier, and then we would need to also know the UV extinction coefficient of the protein and the UV extinction coefficient of the carbohydrate, which actually tends to be very close to 0. So here's an example of how, when we use this method, this was looking at a protein, and it was known from the sequence that it was about 45 kilodalton protein, and with non-reducing native gel, this was consistent with the sample being a monomer in solution. But when SEC was done, the protein was running round about between 200 and 500 kilodalton protein based on standards being run on that column. You can see the red trace is the unknown sample of this material, SLAM F9. So what we did was to take this second peak, take a cut as well we could to try and avoid this larger material, ran it out on SEC moles, and this is kind of what we see here in the green, the stock concentration, and then here a dilution of 1 in 10 of this stock. And you can see that the mass determined from using scattering is constant for both different concentrations. So the lack of concentration dependence supported the idea that this was a monomer, and the evaluated mass was around about 60 kilodaltons. However, we knew that the sequence mass was 46, and so the discrepancy here is too large for an error for the technique typically. And so we went on to apply this conjugate method for determining individually the contributions of a proposed modifier, in this case carbohydrate, and the protein. So doing that, we're able to determine the contribution of the protein component here, and you can see it's very close to the mass of the monomer expected. This is the mass of the modifier on the material, on the particle, and then this is the total mass here corresponding to our initial observation around 60 kilodaltons. Here's another example of the same approach, but this time using detergent, and we were looking at this particular 5-protein complex, which was being solubilized with a detergent called digitonin, and the expected mass of a 1-to-1 complex for this 5-component system was around 170 kilodalton. And again, using the conjugate approach, we were able to determine the mass of the protein in the particles, the mass of the modifier, and then the total observed mass. And here you can see, obviously, there's a lot more detergent on the particle than there was, or the contribution of that modifier is a lot higher than for the glycosylation example. So earlier on, we looked at this equation, and I mentioned that there's this term called the second virial coefficient. And I said at the time that typically we can ignore it. So is this always the case? Well, The second virial coefficient is the thermodynamic property of proteins, and it's the second virial expansion of osmotic pressure with respect to protein concentration. That doesn't really tell you too much, but what it really means is that if the second virial coefficient is positive, then proteins are preferring to interact with the solvent rather than with other protein molecules, and vice versa for a negative value. If we wanted to measure the second virial coefficient, we can do this again with light scattering. What we need to do is to measure the concentration dependence of scattering in a Zim plot. And a Zim plot is essentially a three-dimensional Debye plot where we have run a series of concentrations of our particle of interest, plotted them out as a 3D form then. And then the slope here is equal to the second virial coefficient. And it has been proposed that conditions giving a negative second virial coefficient are considered good for crystal growth because the protein prefers to interact with itself rather than the solvent. And it has, some years ago, it was proposed that if you had a negative value, these were the kind of conditions you should be searching for in terms of crystallization conditions. If you don't know what the second virial coefficient is, you can measure it, but usually at concentrations we're going to be working at in SEC moles, this term in the which the second virial coefficient occurs in is going to be very much less than 1. So this term in the square bracket will approximate to 1. And you can see here that typically for this particular system, and this is typical of many, that the second virial coefficient is on the order of 10 to the minus 4. And therefore, if we work at about 0.1 mg per mil, remember, if we inject sample on to an SEC column at 1 mg per mil, it may well end up at around about 0.1 mg per mil during the chromatography because of dilution on the column. The molecular weight is, say, 50 kilodaltons, and therefore this A2 containing term becomes 1 to the minus 3. If you want to do measurements at very high concentrations, which might be of interest, say, for formulation type of approaches, then you might need to determine the second virial. So coming on to a related technique, dynamic light scattering. This is known by the Wyatt company for some reason as quasi-elastic light scattering, and it's also sometimes referred to as autocorrelation spectroscopy, but most commonly known as dynamic light scattering. So particles that diffuse within some sort of illuminated volume will cause fluctuations in the scattering intensity. And when we're doing static light scattering, we're averaging out those fluctuations by integration. So our signal that we've used in so far for mass determination corresponds to the average in the fluctuations of these signals. But the fluctuations will have different frequencies depending on the particle size. And this is very analogous to fluctuation correlation spectroscopy that I talked about in context of single molecule measurement in talk 6, but using scattered light and certainly not working at single molecule concentrations, and also using scattered laser light rather than fluorescence, which we're using at single molecule level. So the fluctuations are analyzed in a similar manner to FCS, using autocorrelation analysis. In this, you just look at self-similarity of a signal by doing regression with an increasingly offset signal of the same. You collect the signal, you generate a copy of that signal, apply increasing temporal offsets, and regress for each step in time. And you're looking at the persistence of signal in terms of fluctuations. And just as in FCS, we're able to extract the translational diffusion coefficient here, dt. This dt can be input to the Stokes-Einstein equation, and therefore it can yield the hydrodynamic radius of our particle, providing we know the solvent viscosity and obviously the temperature of measurement. And the hydrodynamic radius is not a radius of our particle, but it's equivalent to the radius of a sphere that would diffuse with the same translational diffusion coefficient as our particle. So it doesn't mean our protein is spherical, although they often are quasi-spherical, and it's definitely not a physical dimension of the particle, because it will include some contribution from solvent that is somewhat immobilized or carried with the protein. So how do you measure it? Again, we can measure it in a batch method, which would just be involving using a cuvette of some type. We would put the material in and measure, or it can even be done using a plate reading format, which is very good for screening a wide variety of conditions. Batch methods, these are very sensitive again to small amounts of large particles, just as our static light scattering was. And very complex analysis of the autocorrelation function is required to deconvolve the system if you have a mixed population, which you inevitably will if you do a batch measurement, because even the cleanest and happiest protein will have some small levels of aggregate or large particles present, and these will contribute disproportionately to the fluctuations that you're measuring. So once again, it would be desirable if we could fractionate the material, and that's exactly what we can also do. So in the light scattering detector from Wyatt, one of the static light scattering detectors is removed, and this is then replaced with a dynamic light scattering detector that measures the fluctuations and does the autocorrelation. Because the material is fractionated, we get extremely clean autocorrelation functions and fairly well-determined hydrodynamic radii. What do we use this for? Well, as I've said, the hydrodynamic radius is not a physical radius of our particle, but we can ask questions like, is it typical for what we expect for this protein? So most proteins are compact globular spheres, and under that condition, then the RH, the hydrodynamic radius, scales quite nicely with the size of the system, particularly for smaller proteins. And unfolded or partially disordered proteins will tend to increase the hydrodynamic radius because they diffuse not as a sphere, but as some sort of extended structure. So in this plot here, you can see a red line, which corresponds to what we imagine native proteins will do as a function of just the size of the protein, the number of residues. And this is relating the hydrodynamic radius that we expect to the size of the protein. And if you look at the red dots, you can see, particularly for smaller proteins, they lie very close to this line. The blue line is what we expect for a denatured protein, some sort of random coil, and the translational diffusion that we would see for that. And you can see here where we've managed to measure some proteins which are denatured, they also lie very closely on the line. So when we measure proteins of interest, we can have a look and just see, well, is this what a hydrodynamic radius that we expect for a globular protein? Here's an example where we looked at P53. When you look at this core tetramization domain, which is clearly globular, you see that it lies very close to the native line. If you then add on these extended regions, linker regions, you see that the material then has a much larger hydrodynamic radius than we would expect. This point has a much larger hydrodynamic radius than we would expect for a protein of that size. And then even when you add the folded core, so this is the tetramerization domain, the linker, and this core, it's again a much larger particle than we'd expect. Here's the full length protein, including all of the material. So you can see that it gives you a kind of an idea about what the conformation of your material is like, and it certainly tells you when it isn't a typical globular protein. The other thing we can do with hydrodynamic radius is, for globular proteins, there's a very good correlation between the measured radius of gyration, which if you have a structure, you can determine, you can calculate the radius of gyration from a high resolution three-dimensional structure. And there's a very good relation between the ratio of this and the hydrodynamic radius, and this should be around about 0.8. So here you can see for myoglobin, the radius of gyration is 1.8 nanometers, the hydrodynamic radius is 2.2 nanometers, but just to point out that the dimensions of myoglobin are certainly larger than we're observing for these radii. Why is that useful? Because then when we start to look at other systems, which we want to say, are these globular or not, then we can consider this ratio between the radius of gyration and the hydrodynamic radius. This is a case looking at this protein that comes from the centriole. And in this case, Mark van Breugel, who used to work at the LMB, made mutants in these head domains to stabilize this non-polymerizing structure. And then we were interested in looking at what is the structure of this coiled coil relative to these head domains in solution. We were able to determine with a long construct, this is a crystal structure of a construct that ends at residue 179. But when we were looking in solution, we extended the sequence boundaries by another 150 residues or so. So it's a much larger and more asymmetric particle than this. We measured the radius of gyration close to our limit of 10 nanometers. In fact, about 9, and the hydrodynamic radius was 5.5. This gives us a ratio, not of 0.8, but of 1.6. And this is completely consistent with an extended rod-like shape in solution. So in just in 1 minute or 2 minutes, I just want to flag up a couple of uses for light scattering that is slightly less analytical, but might be useful. One is obviously determining whether you need to make up some fresh buffer, because if you have a lot of grot in your buffer, then you'll see scattering from a laser pointer. And that's obviously not a particularly important application. But for example, one thing just to point out is that scattering will lead to a reduction in transmitted light. That's because some of the light that we expect to pass through a sample is not reaching a detector, for example, in a transmission measurement to measure absorbance. Therefore, we're measuring a reduced level of signal because of this scattering. And if you remember, in this equation, we have wavelength raised to the power 4. This means that the scattering is greatly enhanced at low wavelengths, where we might want to determine the concentration of our protein. So we know that in proteins, the aromatic groups do not really absorb above 320 nanometers, even tryptophan. And so therefore, we know that any absorbance that we see above 320 nanometers is likely to come as a result of this apparent reduction in transmission caused by scattering. And so it's very important that when you measure the protein concentration, you measure the whole absorbance spectrum, go right out to 600 nanometers, and definitely include wavelengths above 320 nanometers where there's no absorbance. If you then graph out the measurement as a function of log of absorbance against wavelength, you can fit this to a linear function, because this wavelength to the power 4 will turn the scattering into a quasi-linear function. You can then extrapolate the apparent absorbance to 280, and then this is what you measured. This is coming from scattering. You should take this value away from this to determine the most accurate protein concentration you can. This is a very small correction, but often the correction is very big. Finally, one more thing to say, that in a fluorimeter, where you're often measuring light fluorescence emission at 90 degrees, you can also use that to measure scattering. And in the case of a system which is forming particles, it's quite useful to apply this to follow kinetics. In this case, the polymerization of filaments. And you can look at the rate of polymerization, you look at the end point, and also you can measure lags which are occurring where there's a delay to formation of sufficiently large filaments. And obviously not forgetting a number of other techniques that are essentially scattering based. We have iSCAT here, which is interferometric scattering, and mass photometry, so-called. I described that in talk 6. Small angle X-ray scattering, Dom mentioned that on Tuesday. You can still do that at synchrotrons, and obviously X-ray diffraction is also a form of scattering. So thanks for listening. Here's a summary of kind of what I've said. I'll let you read through that. And if you have any questions, please put them in the Q&A. Okay, so we have a question from Sissy saying, fresh buffers, there should be no light scattering, which means the laser light is visible. Is that correct? No, when you shine, in that picture, you're looking at 90 degrees. You're looking from above on the laser passing through the buffer, and therefore if your buffer is full of grot, you will be able to see the laser beam. When the buffer is not scattering at all, you won't be able to see it. I think that, well, that's certainly what you're asking. I think you probably already knew that. If you're interested in playing around with that, then a really cool thing to do is to get a little bit of milk and put it into water. And that forms a kind of an emulsion of lots of small particles. And that's a good way of visualizing where light beams are going, because it generates a highly scattering solution. So for example, if you come to a spectrometer and you want to visualize how the light is being focused and where it's going within the instrument, you can do that by putting in a sample which has heavy scattering. You'll be able to visualize the beam. Also another question, how do you decide when to use moles and when to use DLS? Okay, well, we're very lucky in that whenever you do SEC moles here at the LMB, you are going to get the DLS data, whether or not you're interested in them or whether or not you ask to collect them, because in our system, the DLS detection is an independent detector that replaces one of the static angles in the light scattering detector. So you don't have to choose if you do SEC moles, and I would also strongly recommend that you do SEC moles, because the DLS is much more interpretable because you fractionated the material up front. However, if you had a different application, such as you wanted to know what buffer my protein is happy in, what buffer does my protein aggregate horribly in, and what buffer is my protein happy in, then you might want to consider doing batch DLS and use it much more qualitative to see, am I getting a very slowly diffusing particle, which is big, or am I getting a smaller particle, a faster diffusing particle, which is small, and use it much more as a sort of binary measurement. And then you could consider using the plate reading DLS, which allows you to scan potentially 384 different conditions on a single plate if you wanted to. Batch DLS, therefore, I'm kind of hinting, is somewhat less quantitative, but it can be very useful in terms of a quantitative, a more qualitative approach, or kind of screening for conditions. Or, for example, things we've done is people have generated vesicles using membranes that they push through. And we just wanted to confirm that the vesicles were the right size, or look at what the dispersity was within the sample. I don't know if that answers your question about deciding which technique to use, but I think, yeah, maybe it does. Okay, and so finally, obviously, from within the LMB, you know, any questions that occur to you later, or you're interested in trying the technique, or you want training, just contact Stephen or myself, and we'll be very happy to help you.